This executive report examines a novel metric, the Bifunctional Bioavailability Index (BBI), which promises to revolutionize the way we design orally bioavailable ligand-directed degraders (LDDs). We will evaluate its potential impact on optimizing LDDs and the benefits of integrating it with leading AI-driven drug design services. By leveraging these tools, we can accelerate the development of this promising class of therapeutics while overcoming their notorious oral bioavailability challenges. 1. Executive summary. A new study proposes a computational metric called the Bifunctional Bioavailability Index (BBI) to predict the oral bioavailability of ligand-directed degraders (LDDs), also known as proteolysis targeting chimeras (PROTACs). The BBI integrates molecular size, polarity, lipophilicity, and molecular shape, which may provide a more accurate assessment of oral bioavailability in LDDs than existing physicochemical descriptors or in vitro cell permeability assays. If validated across various LDD chemistries, this metric holds significant promise for advancing the clinical translation of these complex molecules. In this report, we will investigate the relevance of the BBI for top heterobifunctional degraders currently in clinical trials and identify leading AI-automated drug design services well-positioned to leverage such a tool. 2. Background: the challenge of LDD oral bioavailability. Ligand-directed degraders (LDDs) are engineered heterobifunctional molecules designed to selectively degrade target proteins by recruiting 𝗕𝗥𝗢𝗧𝗔𝗕𝗓 ligases. They hold immense therapeutic potential by offering an alternative approach to traditional small molecule inhibitors or monoclonal antibodies. However, the path to clinical success for LDDs is fraught with challenges, primarily attributed to their large size and unique physicochemical properties. These characteristics deviate from conventional drug-likeness rules, making them more susceptible to premature degradation in biological systems, thereby impacting their oral bioavailability. Achieving adequate oral bioavailability is paramount for patient convenience and broader applicability. Consequently, it remains a critical challenge for the field. 3. Validity of the BBI claim. In the study, the authors assert that BBI can differentiate oral bioavailability in LDDs more effectively than polarity, lipophilicity, or size separately, and is more effective than in vitro cell permeability assays in predicting orally bioavailable LDDs. Unfortunately, specific BBI values for each clinical candidate were not provided in the study, making it challenging to definitively verify these claims without additional data. However, assuming these findings hold true, and the BBI approach is robust and generalizable across different 𝗕3 ligase recruiters and target binding moieties, it could represent a significant advance in overcoming the oral bioavailability hurdle for LDDs. 4. Impact of BBI on clinical stage LDDs. The BBI's potential impact on the development of current clinical stage LDDs is substantial. For instance, the estrogen receptor degrader, Vepdegestrant ARV-471, LSC102, developed by Arvinas in partnership with Pfizer, is currently in phase 3 trials for the treatment of positive breast cancer. Ensuring optimal oral bioavailability for this and future degraders is critical for widespread patient access and convenience. Similarly, Bavdegalutamide ARV-110, ARV-766, another degrader from Arvinas partnered with Pfizer, Novartis for prostate cancer, is in phase 2 trials. The BBI could have aided in the selection of lead compounds with superior oral PK properties, potentially minimizing attrition in later stages due to poor absorption. Additionally, 𝗕𝗦𝗢𝗧𝗬, an IRAK4 degrader developed by Chimera Therapeutics in collaboration with Sanofi for immune-inflammatory diseases such as hidradenitis suppurativa and atopic dermatitis, is also in phase 2 trials. The diversity of targets and disease areas targeted by PROTACs underscores the broad need for effective bioavailability prediction. A metric like BBI could have accelerated the progression of 𝗕𝗦𝗢𝗧𝗬 by flagging molecules with suboptimal oral characteristics early in discovery, streamlining lead optimization. Overall, the BBI's predictive power would have been highly beneficial in the development of these leading clinical candidates. For instance, the BBI could have aided in the early prioritization of compounds with a higher likelihood of achieving oral bioavailability, reducing costly in vitro and in vivo testing on less promising candidates. Furthermore, the BBI could have guided medicinal chemists in modifying linker chemistry, E3 ligase binders, and target-engaging moieties to improve oral absorption while maintaining target degradation. By addressing this key attrition factor, oral pharmacokinetics, earlier in the drug discovery pipeline, the BBI could have helped mitigate risks and accelerated the development of safe and effective LDD-based therapies for various diseases. 5. Leading AI automated drug design services. For LDDs, we have identified the top 3 global AI automated services well-positioned to integrate and leverage such a metric: 𝗕𝗥𝗢𝗧𝗔𝗕𝗜𝗄𝗧 and 𝗕𝗥𝗢𝗧𝗥𝗢𝗧𝗏. 𝗕𝗥𝗢𝗢𝗧𝗔𝗕𝗜𝗄𝗧 is a pioneer in AI drug discovery and automated research. Their platforms, including 𝗕𝗜𝗢𝗧𝗔𝗕𝗜𝗄𝗧 and 𝗕𝗏𝗔𝗠, are designed for generative chemistry and lead optimization. Integrating BBI into their algorithms would allow them to generate novel LDDs with favorable bioavailability. 𝗕𝗏𝗡𝗡𝗡 specializes in AI-powered structure-based drug design. Their 𝗕𝗏𝗡 platform excels at virtual screening and identifying novel hits. Integrating BBI would enable them to prioritize potential LDD candidates with a higher likelihood of oral bioavailability, significantly enhancing their lead identification process. 𝗕𝗥𝗢𝗧 combines generative AI, such as their 𝗕𝗥𝗢𝗡 platform for de novo design, with AI-driven retrosynthesis, making their SPIA platform ideal for integrating BBI. Their molecular odyssey aims for fully autonomous labs. BBI could be integrated directly into 𝗕𝗥𝗢𝗡𝗧's multiparametric optimization capabilities, guiding the generative AI to design LDDs with favorable BBI scores. Subsequently, this would streamline automated synthesis and testing phases. These platforms, by incorporating the BBI, could collectively advance our ability to design, optimize, and validate orally bioavailable LDDs, accelerating the development of much-needed therapies for various diseases. 6. Conclusion and recommendations. The introduction of the BBI represents a promising development in addressing a major obstacle for LDDs, oral bioavailability. Its effectiveness in predicting this crucial property surpasses existing methods, paving the way for more efficient discovery and development of these complex therapeutics. For current clinical stage LDDs, the BBI's predictive power would have been invaluable. Looking ahead, we strongly recommend strategic actions. Firstly, internal validation and adoption. Explore avenues for internal validation of the BBI concept using our own LDD datasets and computational models. Upon validation, prioritize its adoption as a key metric in our early-stage LDD design and lead optimization workflows. Secondly, strategic partnerships with AI platforms. Engage with leading AI automated drug design service providers, such as 𝗕𝗥𝗢𝗢𝗧𝗔𝗕𝗜𝗄𝗧, atomize, and 𝗕𝗥𝗢𝗧𝗔𝗕𝗜𝗄𝗧 to explore collaborations focused on integrating BBI as a core parameter within their generative and predictive AI models for LDD discovery. This will allow us to harness cutting-edge computational power to design and optimize orally bioavailable PROTACs more efficiently. Moreover, continued research into novel descriptors and computational models that further refine the prediction of complex physicochemical properties for LDDs should be supported. Building upon the foundation laid by the BBI, in conclusion, the BBI offers a beacon of hope in the pursuit of developing effective and accessible LDD-based therapies. By embracing this innovative metric and strategic collaborations, we can collectively move closer to realizing the full potential of this groundbreaking technology for the benefit of patients worldwide.